Hello and welcome dear students to the massive open online course on human genetics. Today's topic of deliberation is basics of DNA replication. The main objectives of today's lecture are to study about the basic idea of DNA replication, to know about the various enzymes involved in DNA replication, to discuss the mechanism of DNA replication, and finally, we will discuss the critical steps involved in the process of DNA replication. Dear students, first of all, we will start with the introduction to DNA replication. The cell cycle or cell division cycle is the series of events that take place in a cell that cause it to divide into two daughter cells. It is one of the most remarkable and important processes that is happening within any living organism. Cell division plays an important role in all living organisms as it is essential for growth, repair, and reproduction. Cells divide to allow multicellular organisms to grow, to reproduce, and create identical copies of themselves and to replace or repair damaged or dead cells in multicellular organisms. Also, single-celled organisms need to go through cellular division to reproduce. Before a cell decides, it must first copy or replicate its entire genome so that each cell resulting into daughter cell ends up with its own complete genome. Replication is an essential process because whenever a cell divides, the two new daughter cells must contain the same genetic information or DNA as the parent cell. DNA replication is the process by which the genomic DNA of an organism is copied in cells. The replication process relies on the fact that each strand of DNA can serve as a template for duplication. DNA replication results in the duplication of DNA content in the cell, which is essential to maintain the genetic continuity in the newly formed cells during cell division. Each cell receives the exact copy of DNA as that of the parent cell. This is required for growth and maintenance. DNA replication is one of the most amazing tricks that DNA does. If you think about it, each cell contains the entire DNA you need to make the other cells, and we start out from a single cell and we end up with trillions of cells. And during that process of cell division, all of the information contained in that DNA, that is the genetic material in a cell, has to be copied, and it has to be copied perfectly without any errors. Hence, DNA during replication process is a molecule that can be replicated to make almost perfect copies of itself, which is all the more amazing considering that there are almost 3 billion base pairs of DNA in a human being, all of which must be accurately copied when any one of our trillions of cells divides in our body. The basic mechanisms of DNA replication are similar across organisms like humans and other eukaryotes. DNA replication is semi-conservative, meaning that each strand in the DNA double helix acts as a template for the synthesis of a new complementary strand. Cells need to copy their DNA very quickly and accurately with very few errors. To do so, they use a variety of enzymes and proteins which work together to make sure DNA replication is performed smoothly and accurately. Replication uses specific enzymes like DNA polymerases, which are molecules specifically dedicated to just copying DNA. Replicating the entire DNA in a single human takes several hours of just pure copying time. At the end of this process, once the DNA is all replicated, the cell actually has twice the amount of DNA that it needs, and the cells can then divide and parcel this DNA into the daughter cell, so that the daughter cell and the parental cell in many cases are absolutely identical. Before going to the detailed process of DNA replication, let us discuss the key points of DNA replication. DNA replication is semi-conservative in nature. Each strand of DNA double helix acts as a template for synthesis of a new complementary strand. New DNA strand is synthesized by special enzymes called DNA polymerases, which require a template and a primer that is a starter and synthesize DNA in the 5' to 3' direction. In DNA replication process, one new strand, the leading strand, is made as a continuous piece, while as the other, that is the lagging strand, is made in small pieces known as Okazaki fragments, named for the Japanese scientist who discovered them. The leading strand can be extended from one primer alone, whereas the lagging strand needs a new primer for each of the short Okazaki fragments. In addition to DNA polymerases, the replication of DNA requires other enzymes including DNA primase, DNA helicase, DNA ligase, and topoisomerase. Dear students, let's now discuss the DNA replication in detail. By definition, DNA replication is the process of producing two identical copies of DNA from one original DNA molecule. As we all know that DNA is made up of millions of nucleotides which are composed of deoxyribose sugar, phosphate, and nitrogenous base. The complementary pairing of these bases keeps the double strand intact. So, to make two copies of one DNA, these hydrogen bonds in between the bases should be broken to begin the replication. DNA replication is semi-conservative, meaning that each strand in the DNA acts as a template for the synthesis of a new complementary strand. Semi-conservative because once DNA molecule is synthesized, it has one old strand from the parent and the other strand is a newly formed strand. The process of replication starts by taking one DNA molecule and giving two daughter molecules, with each newly synthesized molecule containing one new strand and one old strand. DNA replication simply is the process by which a DNA makes a copy of itself. Though easy as it may sound, it's a complex process happening inside our cells, and many enzymes, proteins, and metal ions should work coherently together to make this process happen. Replication follows several steps that involve multiple proteins called the replication enzymes and RNA. In eukaryotic cells such as animal cells and plant cells, DNA replication occurs in the S phase of the interphase during the cell cycle. The process of DNA replication is vital for cell growth, repair, and reproduction in organisms. Dear students, we now know that DNA is a self-replicating structure and it replicates semi-conservatively. However, DNA replication is catalyzed by a set of enzymes. Let's learn about the DNA replication process and the role of each enzyme involved in DNA replication. Let's first discuss about the enzymes involved in the DNA replication process. First is nucleases. A nuclease is an enzyme that can cleave the phosphodiester bonds present in between the nucleotides. On the basis where they cleave, they are characterized as exo and endonucleases. Exonucleases cleave nucleotides from their respective ends. Corresponding to this fact, these exonucleases show activity from both directions, that is 5' direction to 3' direction and 3' to 5' direction. Endonucleases, on the other hand, act on the region in the middle of the targeted nucleotide. Endonucleases are selective to which molecule that cleave and are subdivided as DNase for DNA for cleaving and RNase for RNA cleaving. Moreover, recently discovered nucleases are also being used for gene editing, such as Cas9 in the CRISPR genome editing technique. Dear students, now let us discuss the restrictive endonucleases or restriction enzymes. These are the ones that cleave DNA into fragments at or near the specific recognition sites within the molecule known as restriction site. To cleave, the DNA restriction endonucleases make two incisions, once through each sugar phosphate backbone of the DNA double helix. These endonucleases recognize a specific sequence of nucleotide and produce a double-stranded cut in the DNA. This specific sequence is usually 4 to 8 bases long and is present in the recognition site. Dear students, let's now discuss DNA polymerases, the other important enzymes involved in DNA replication. DNA polymerases are the enzymes that are responsible for adding new nucleotides and synthesizing a new strand of DNA by taking the old fragmented strand as a template. DNA polymerases also possess exonuclease activity that cuts incorrectly added nucleotides and allows the DNA replication to happen without the errors. These enzymes are essential for DNA replication and usually work in groups to create two identical duplexes from a single original DNA duplex. All organisms, either unicellular or multicellular, that use DNA as their genetic information require one or more DNA polymerases for their survival. Most DNA viruses encode their own DNA polymerase, while a few predate those of their infected cells for replicating their genomes. Hence, DNA polymerases are essential for all kingdoms of life, from viruses to humans. DNA polymerase is of many types and functions are based on the cell they are found in. In prokaryotic cells, there are 3 DNA polymerases: DNA polymerase 1, DNA polymerase 2, and DNA polymerase 3. DNA polymerase 1 is a Rapier polymerase with 5' to 3' and 3' to 5' exonuclease activity. It is involved in the processing of Okazaki fragments during lagging strand synthesis. DNA polymerase 2 has 3' to 5' exonuclease activity and participates in DNA Rapier with 5' to 3' polymerase activity. DNA polymerase 3 is the primary enzyme involved in the DNA replication of Escherichia coli. It has 3' to 5' exonuclease activity and 5' to 3' polymerase activity. In eukaryotic cells, there are 5 DNA polymerases: DNA polymerase alpha, DNA polymerase beta, DNA polymerase gamma, DNA polymerase delta, and DNA polymerase epsilon. So DNA polymerase alpha is a Rapier polymerase with 3' to 5' exonuclease activity and 5' to 3' polymerase activity. On the other hand, DNA polymerase beta is a Rapier polymerase. DNA polymerase gamma shows polymerase activity from 5' to 3' in and exonuclease activity from 3' to 5' in. It is involved in mitochondrial DNA replication. Now, DNA polymerase delta, it shows the 3' to 5' exonuclease activity and 5' to 3' polymerase activity. This enzyme is involved in lagging strand synthesis. The DNA polymerase epsilon shows 3' to 5' and 5' to 3' exonuclease activity. This enzyme not only Rapiers but also synthesizes the leading strand efficiently in a 5' to 3' direction. It is the prime enzyme involved in DNA replication. Dear students, finally, the DNA polymerases, which are multi-subunit enzymes including polymerase alpha, polymerase delta, and polymerase epsilon, are critical for the accurate replication of cellular DNA. While polymerase alpha initiates DNA synthesis, polymerase delta and polymerase epsilon perform the majority of the DNA replication, with Pol delta synthesizing the lagging strand and Pol epsilon synthesizing the leading strand. Given the importance of accurate DNA replication, the proper function of these enzymes is critical to maintain DNA stability. Dear students, now we will discuss DNA ligases, another important class of enzyme involved in DNA replication. DNA ligase is a specific type of enzyme that facilitates the joining of DNA strands together by catalyzing the formation of a phosphodiester bond. This enzyme joins 3-hydroxyl group of one nucleotide with 5-phosphate end of another nucleotide at an expense of ATP. DNA ligases utilize either ATP or NAD positive as the nucleotide cofactor. It plays a role in repairing single-strand breaks in duplex DNA in living organisms, but some forms may specifically repair double-stranded breaks. DNA ligases, together with RNA ligases and mRNA capping enzymes, constitute the nucleotidyl transferase superfamily of enzymes. Dear students, let's now discuss the DNA helicase enzyme, another important enzyme involved in the DNA replication. DNA helicase is a motor protein that moves directionally along a nucleic acid phosphodiester backbone, separating two nucleotides of DNA molecule. They separate double-stranded DNA molecules into single strands, allowing each strand to be copied. During DNA replication, this DNA helicase unwinds DNA at the origin, a site where the replication is to be initiated or started. DNA helicase continues to unwind the double helix of DNA and thus forms a structure called replication fork, named after the forked appearance of two strands of DNA when unzipped apart. It is an energy-driven process as it involves the breaking of hydrogen bonds between annealed nucleotide bases. Now let us discuss the DNA primase. DNA primase is an enzyme that is capable to synthesize short stretches of RNA molecules or sequences known as primer. Primers are an integral part of DNA replication. These primers serve as the initiating site for the addition of nucleotide by DNA polymerase. DNA polymerase can only add nucleotide at pre-existing 3-hydroxyl groups, which is thus provided by the primers. As we can see that primers are short stretches of RNA, but replication is of DNA. So therefore, after elongation of the chains of nucleotides, these primers are replaced by DNA. Dear students, now let us discuss the enzyme DNA topoisomerase. DNA topoisomerase is a class of enzymes that release helical tension during transcription and replication by creating transient nicks within the phosphate backbone on one or both strands of the DNA. This tension is aroused when the DNA molecule unwinds due to helicase activity and forms a replication fork. The progress of the replication fork generates supercoils, making it hard for other machinery involved to access the DNA molecule. These are subdivided into two categories: class 1 DNA topoisomerase and class 2 DNA topoisomerase. Class 1 DNA topoisomerase makes a single-stranded break to relax the helix and progress the process. Class 2 DNA topoisomerase break both the strands of DNA helix. This class of topoisomerases are also very important during the cell cycle for the condensation of chromosomes. Dear students, let's now discuss about single-stranded binding protein, that is SSBP. The single-strand binding protein are DNA binding proteins that bind to single-strand DNA to facilitate DNA replication. Single-strand binding protein prevent the hardening of strands during DNA replication. It also protects strands from nucleus degradation and prevents the rewinding of the DNA. These proteins destabilize helical duplexes so that DNA polymerase can hold onto the DNA during DNA replication, recombination, and repair. It also removes unwanted secondary structures on strands for easy access of the strands to the machinery involved in DNA replication. Thus, single-strand binding proteins stabilize the single-stranded DNA structure that is important for genomic progression. So in summary, here are the list of 7 enzymes and proteins used in DNA replication. First, the nucleases. Second, polymerase. Third, DNA ligase. Fourth, DNA helicase. 5, DNA primase. 6, DNA topoisomerase. And the finally 7th one is the single strand binding protein or SSB. Dear students, now let us discuss DNA replication in detail. DNA replication takes place in 3 major steps. First is the opening of the double-stranded helical structure of DNA and separation of the strands. Second is the priming of the template strands. Third is the assembly of the newly formed DNA segments. During the separation of DNA, the 2 strands uncoil at a specific site known as origin of replication. Replication always starts at a specific location on the DNA, which are called origins of replication and are recognized by their sequence. Specialized proteins recognize the origin, bind to the site, and open up the DNA. As the DNA opens, 2 Y-shaped structures called replication forks are formed, together making up what's called a replication bubble. The replication forks will move in opposite directions as the replication proceeds. Helicase is the first replication enzyme to load at the origin of replication. Helicase's job is to move the replication forks forward in opposite directions by unwinding the DNA, that is breaking the hydrogen bonds between the nitrogenous bases of the 2 ladders or 2 strands of the DNA. Proteins called single strand binding proteins coat the separated strands of the DNA near the replication fork, keeping them from coming back together into a double helix. With the involvement of several enzymes and proteins that prepare or prime the strands for duplication. At the end of the process, DNA polymerase enzyme starts to organize the assembly of the new DNA strand. DNA polymerases can only add nucleotides to the 3' end of an existing DNA strand. They use the free hydroxyl group found at the 3' end as a hook, adding a nucleotide to this group in the polymerization reaction. How then does DNA polymerase add the first nucleotide at a new replication fork? Alone it cannot. So then the problem is solved with the help of an enzyme called the primase. Primase makes an RNA primer or short stretch of nucleic acid complementary to the template that provides a 3' end for DNA polymerase to work on. A typical primer is about 5 to 10 nucleotides long. The primer primes DNA synthesis that it gets started. DNA replication is one of the most essential mechanisms of a cell's function, and therefore intensive research has been done to understand its processes. The mechanism of DNA replication is well understood in Escherichia coli, which is also similar to that in eukaryotic cells. In E. coli, DNA replication is initiated at the Ori-Clox or Ori-C point to which DNA A proteins bind while hydrolysis of ATP takes place. Dear students, let's now discuss the 3 important steps involved in DNA replication: the initiation, elongation, and termination. DNA initiation is the stage where DNA replication is initiated. DNA synthesis is initiated within the template strand at a specific coding region site known as the origin site. The origin sites are targeted by the enzymes which recruit additional proteins that help in the replication process to form a replication complex around the DNA origin. There are several origin sites on which DNA replication is initiated, and they are all known as replication sites. The formed replication complex contains the DNA helicase enzyme whose function is to unwind the double helix, exposing the 2 strands which act as templates for replication. The mechanism of DNA helicase enzyme is by hydrolyzing the ATP that is used to form the bonds between the nucleobases, thus breaking the bond that holds the 2 strands together. Additionally, during initiation, DNA primase enzymes synthesize small RNA primers that kickstart the function of DNA polymerase. DNA polymerase enzyme functions by growing the new DNA daughter strand. Dear students, let's now discuss elongation. This is the phase where the DNA polymerase grows the new DNA daughter strand by attaching to the original unzipped template strand and the initiating short RNA primer. The DNA polymerase is able to synthesize a new strand that matches the template by extending the primer via the addition of the free nucleotides to the 3' end of the DNA. One of the template reads in the 3' to 5' direction, and therefore the DNA polymerase synthesizes the new strand in the 5' to 3' direction, which is known as the leading strand. Along the template strand, DNA primase synthesizes a short primer or short RNA primer at the beginning of the template in the 5' to 3' direction, which initiates the DNA polymerase to continue synthesizing new nucleotides, extending the new DNA strand. The other template, 5' to 3' is elongated in an anti-parallel direction by the addition of short RNA primers, which are filled with other joining fragments, forming the newly synthesized small strands. These short fragments are known as Okazaki fragments. The synthesis of the lagging strand is discontinuous since the newly formed strand is disjointed. The RNA nucleotides from the short RNA primer must be removed and replaced by DNA nucleotides, which are then joined together by the DNA ligase enzyme. Dear students, let's now discuss termination. After the synthesis and extension of both the continuous and discontinued strands, an enzyme known as exonuclease removes all RNA primers from the original strands. The primers are replaced with the right nucleotide bases while removing the primers. Another type of exonuclease proofreads the newly synthesized strain, checking, removing and replacing any errors formed during the synthesis of these small fragments. So, DNA ligase enzyme joins these small Okazaki fragments to form a single unified strain. The ends of the parent strain consist of a repetition of DNA sequences known as telomeres, which act as protective caps at the ends of chromosomes, preventing the fusion of nearby chromosomes. The telomeres are synthesized by a special type of DNA polymerase enzyme known as telomerase. It catalyzes the telomere sequences at the end of the DNA. On completing or completion, the parent and complementary strand coil into a double helical shape, producing two DNA molecules, each passing one strand from the parent molecule and one new strand. Dear students, let's now discuss briefly about the Okazaki fragments and their significant role in DNA replication. The two DNA strands run in opposite or anti-parallel direction, and therefore to continuously synthesize the two new strands at the replication fork requires that one strand is synthesized in 5' to 3' direction, while the other is synthesized in the opposite direction, that is 3' to 5' direction. However, DNA polymerase can only catalyze the polymerization of the DNTPs in 5' to 3' direction. This means that the other opposite new strand is synthesized differently. But how? By the joining of discontinuous small pieces of DNA by DNA ligases that are synthesized backward from the direction of movements of the replication fork. These small pieces or fragments of the new DNA strand are known as the Okazaki fragments. The Okazaki fragments are then joined by the action of DNA ligase, which form an intact new DNA strand known as the lagging strand. The lagging phase is not synthesized by the primer that initiates the synthesis of the leading strand. Instead, a short fragment of RNA serves as a primer, that is RNA primer, for the initiation of replication of the lagging strand. RNA primers are formed during the synthesis of RNA, which is initiated de novo, and an enzyme known as primase synthesizes these short fragments of RNA, which are 3 to 10 nucleotides long and complementary to the lagging strand. The Okazaki fragments are then synthesized by the extension of the RNA primers by DNA polymerase. However, the newly synthesized lagging strand is that it contains an RNA-DNA joint, defining the critical role of RNA in DNA replication. Now we will learn about the significance of replication fork and the essence of its function in replication of DNA. The replication fork is the site of active DNA synthesis, where the DNA helix unwinds and single strands of the DNA replicate. Several sites of origin represents the replication forks. The replication fork is formed during DNA strand unwinding by the helicase enzyme, which exposes the origin of replication. A short RNA primer is synthesized by primase and elongation done by DNA polymerase. The replication fork moves in the direction of the new strand synthesis. The DNA strands are synthesized in two orientations, that is 3' to 5' direction, which is the leading strand, and 5 to 3' orientation, which is the lagging strand. The two sides of the new DNA strands, leading and lagging strands, are replicated in two opposite directions from the replication fork. Therefore, the replication fork is bidirectional. The leading strand is the new DNA strand that is continuously synthesized by the DNA polymerase enzyme. It is the simplest strand that is synthesized during replication. The synthesis starts after the DNA strand has unzipped and separated. This generates a short piece of RNA known as RNA primer by the DNA primase enzyme. The primer binds to the 3' end of the strand, thus initiating the synthesis of the new strand, that is the leading strand. The synthesis of the leading strand is a continuous process. This is the template strand, that is 5' to 3' direction, that is synthesized in a discontinuous manner by RNA primers. During the synthesis of the leading strand, it exposes small short strands or template that are then used for the synthesis of the Okazaki fragments. The Okazaki fragments synthesize the lagging strand by the activity of DNA polymerase, which adds the pieces of DNA, that is the Okazaki fragments, to the strands between the primers. The formation of the lagging strand is a discontinuous process because the newly formed strand, lagging strand, is the fragmentation of short DNA strands. Dear students, let's now discuss the main differences in the process of replication of DNA in both prokaryotes and eukaryotes. DNA replication process in prokaryotes. The DNA replication in prokaryotes takes place in the manner that is the two strands of DNA unwind at the origin of replication, helicase opens the DNA, and replication forks are formed. The DNA is coated by the single strand binding proteins around the replication fork to prevent rewinding of the DNA. Topoisomerases then prevent the supercoiling of DNA. RNA primers, on the other hand side, are synthesized by primase. These primers are complementary to the DNA strand. So DNA polymerase III starts adding new nucleotides at the end of the primers. The leading and lagging strands continue to elongate. The primers are removed and the gaps are filled with DNA polymerase I and sealed by the DNA ligase. Dear students, the DNA replication in eukaryotes. The DNA replication in eukaryotes is similar to the DNA replication in prokaryotes. However, the initiation process is more complex in eukaryotes than in prokaryotes. In eukaryotes, there are multiple origins of replication present. A pre-replication complex is made with the other initiator proteins. The process is entirely the same, but the enzymes used are different. For example, in eukaryotes, the polymerization process is carried out by the enzyme Pol delta, whereas in prokaryotes, it is done by DNA Pol III. Dear students, it was all about today's lecture. Hope you have understood it well. See you with a new topic next time. Take care. Goodbye.